Hi class, we're going to do chapter 15 with the screencast-o-matic. It's our shortest chapter, so hopefully within two 15-minute screencast-o-matics, I can get through the chapter. You should have your study guide in front of you. It's just a 2-page study guide this time, and I'm also, to save time, going to skip some of the stories that I usually talk about. So, skipping through the stories. The first thing we're going to do is review some of what was in chapter 14. So if you turn to page 2 of your study guide, this is just a review. Now you do have this picture on your study guide, but I am not going to ask you to fill this out. So repeat, I am not going to make you fill this out. This is just a review that we would usually do in class of chapter 14. So the things I want you to know are, again, the 4 eras: Precambrian, Paleozoic, Mesozoic, and Cenozoic. How old is the Earth? 4.5 to 4.6 billion years old. How old are prokaryotes? Prokaryotes appeared 3.5 billion years ago, so they're happening in the Precambrian era. And again, on this slide, I'm showing you the events that occurred. You can see up on the very top, you can see the entire geological time scale, and then these numbers are appearing where they happened in that geological time frame. So I told you in the chapter 14 recording that I would be doing a matching for the ages. So if you can know for Precambrian these 4 points, so the Earth was formed, prokaryotes appeared 3.5 billion years ago. So again, it took 1 billion years before the Earth had its first life. And then oxygen production began about 2.5 billion years ago. So it took another 1 billion years before oxygen was produced. And then single-celled eukaryotic organisms evolved 2.2 billion years ago. So continuing with this, we've got multicellular organisms appearing at number 5. Again, this is in the Precambrian era. They evolved about 1 billion years ago. And then at the end of the Precambrian, we have all the major phyla of animals. This was known as the Cambrian explosion, which began about 540 million years ago. Number 7, you see that happening in the Paleozoic. So you've got 475 million years ago, plants and fungi colonized land, amphibians evolved from fish, vertebrate life moved on to land. Now, how am I going to ask this on the exam? I will give you a fact, and you have to put it in the right era, either Precambrian, Paleozoic, Mesozoic, or Cenozoic. Remember, if I give you the 4 eras out of order, in order to get them in the correct order, you put them in order alphabetically, Cenozoic being now, Mesozoic, Paleozoic, and Precambrian. So here's some major episodes in the history of life. Again, I am not going to have you label this on the exam. Just know in the combination of chapter 14 and chapter 15 study guide on the bottom there, be able to match those things to the correct era. Okay, we don't have this Discovery Channel movie, so moving right along. We're going to talk about how life could have started on the planet Earth. There are several hypotheses out there, but we're going to talk about one. We're just going to talk about one of them. So most biologists think that the chemical and the physical processes were just right on the early Earth for life to start forming. So if you look on the first page of your study guide, you've got the 4-stage hypothesis for the origin of life. This is the only one we're going to talk about. There are others that talk about meteors bringing life to the planet. But even if you go with that hypothesis, how did the life start on the planet where it started? So we're just going to talk about this four-stage hypothesis for the origin of life. So if you look at your first concept question, you've got the abiotic synthesis of organic monomers. So let's look at this one. So you just put stage 1, abiotic synthesis of organic monomers. This was studied extensively by Stanley Miller back in the 1950s, and he devised an experiment that could produce small organic molecules. So if you look, he set up with this other man named Urey, Miller and Urey's famous experiment. They set up this glass contraption and they heated the sea, which was water, so they had water vapor going up, and then they had an atmosphere of methane, ammonia, and hydrogen, and then they would send zaps through it like you would have with lightning, and then those were able to condense and to be cooled, and then he actually saw some of these abiotic, meaning without life, formation of organic monomers. So you can say, check, we've seen this one happening in the lab. The next stage is the abiotic synthesis of polymers. So if you have monomers, can you get them to go into polymers? And researchers have observed polymerization of monomers in various solutions. So check, we have seen this happening in the lab. The third stage is the origin of self-replicating molecules. So this work, some of this work was actually done in Colorado, in Boulder. Laboratory experiments have shown that short RNA molecules can assemble spontaneously. So this stage 3, origin of self-replicating molecules, check. We have seen this happen in the laboratory also. Now, stage 4 is the formation of pre-cells. Now remember, the Earth had billions of years to get this going, and we don't have that huge amount of time in a lab situation. But, so your stage 4 of that first concept question is going to be formation of pre-cells, and then all these things would have to get together. So if we look at that, we have the self-replication of RNA. We've seen that. RNA can act as a template on which a polypeptide forms, and then that polypeptide could act on the RNA. But all of that must happen within a membrane. That's the part that took 1 billion years. So the thing that we have seen are pre-cells forming. When you put polypeptides in a solution or lipids in a solution, you can see circles forming. So over billions of years, could the self-replicating RNA happen within a polypeptide lipid circle? That's the part we have not seen in the lab yet, but the hypothesis is that's how it must have happened, because we don't have billions of years to observe it in the lab. So over millions of years, natural selection would favor the most efficient pre-cells, and then the first prokaryotic cells evolved. So if we look at prokaryotes now, they lived and evolved all alone on Earth for 2 billion years. We actually have fossilized prokaryotes, which is that picture you see on the left. Prokaryotes are everywhere. Remember, prokaryotes are bacteria. They far outnumber the eukaryotes. They can cause disease. They can also be very beneficial. So now we get back to our study guide, and we've got some terms on the first page. we've got halophiles, thermophiles, and methanogens. So the majority of prokaryotes are bacteria, but there are some archaea which are considered extremophiles. So these extremophiles are these three varieties. So halophiles live in extremely salty environments. Extreme thermophiles inhabit very hot water, like you have some archaea, some thermophiles living in Yellowstone National Park in the bubbling, boiling mud vats there. And then we have methanogens, which include the archaea that inhabit the bottoms of lakes and swamps. So this is an example of salt-loving archaea. They have these seawater evaporating ponds at the edge of San Francisco Bay. The seawater starts out with 3% salinity, but as they try to extract the water, these ponds become 15 to 20% salinity. These are the ones that the halophiles love to live in. Okay, so just a reminder from previous chapters, we've got prokaryotic cells lack true nuclei. They have other membrane enclosed, they lack other membrane enclosed organelles, and they have cell walls exterior to their plasma membranes. So if you look on the very bottom of the first page of your study guide, it asks you, what are the three shapes of bacteria? So the three shapes include cocci, these are the round circles, bacilli, these are the rod-shaped, kind of looking like a pickle, and then we have the spirochetes, which are spirals. Now this kind of question is going to appear in a multiple choice question, so see if you could pick out cocci, bacilli, and spirochetes in a multiple choice kind of question. Just a little bit more info. Cocci, if you've ever had strep throat, it's streptococci. Bacilli is going to be, an example of that is E. coli that lives in your intestinal tract. And then spirals, this includes the organism that causes Legionnaire's disease. I would normally tell you a story about that, but I'm just going to move along. So most prokaryotes are unicellular and very small, but some have formed true colonies. Some show specialization of cells. This is the cyanobacteria. These guys were key in the beginnings of Earth to bring oxygen to the planet by photosynthesizing. And then some are also very large, as you can see here. They don't tell us what kind of fly this is, so it's hard to tell the shape, the true size. But here you can get a picture in your mind of how evolution could happen when we've got true colonies, specialization of cells, and some being very large. About half of all prokaryotes are mobile, meaning they can move, and they use flagella. So if you look at your terms, fourth term, hopefully this is review. A flagella is going to be the whip-like tail that helps move the bacteria. And I used this as an example in chapter 14, how flagella started out as a poisonous injector and then evolved gradually into a movable, something that moved. So this brings me to the end of the first 15 minutes, so I'm going to sign off now.